Fantastic. All right. Well, this is a really absolutely wonderful meeting. Fantastic program, really great discussions. Real pleasure to be here. So what I'll do is I'll share some thoughts about how we've sort of been thinking about how protein aggregation and condensates go together. And obviously, my sort of group in Cambridge, we're interested in understanding the different states that proteins occupy very generally. And it turns out, of course, that there's many of these states outside of the canonical sort of folded and unfolded states of proteins. There's membrane-bound states, there's functional complexes. But here, what I'll do is I'll focus largely, as announced in the title, on these two states here. Obviously, the liquid condensate state, which clearly is the focus of the meeting here, and obviously these are functional states, but also I'll be talking about how this state is connected to the aggregated state of proteins, which is also featured in many of the talks yesterday, which, unlike the condensate state, is typically associated with malfunction. And it's interesting, all of the features which make these condensates so useful for biology, the fact that they're dynamic, they're reversible, these are the features which are really lost when proteins go from this liquid state here into this solid state here. And it turns out that there's over 50 or so diseases which are all associated with the uncontrolled formation of these types of structures from proteins, which are normally supposed to be in some other part of the phase space. So generally what we're trying to understand really is the nature of different phases that proteins can occupy and the transitions between these phases. And we're increasingly realizing that there's really very close connections between this liquid state and the solid state. And so to illustrate this, I'll start with one example, which is sort of the archetypical aggregating peptide, the A-beta peptide. So that's the peptide which aggregates in the extracellular space in Alzheimer's disease. And in sort of work over the past 10, 15 years or so, we spent a lot of time in our group and also together with collaborators looking at the molecular steps in this aggregation process. But it turns out that the one step which we didn't sort of really fully get our heads around was the initial primary nucleation. So how did this aggregation process start? And so, of course, based on work from Simon and others, it sounds like a plausible idea that there might be condensates involved in this process in some way. But it turns out that it took many years actually for us to understand how condensation was connected to the aggregation of A-beta, because the peptide itself is not that prone to form liquid compartments. But it turns out that as soon as you put it in the presence of many different kinds of lipids, in particular zwitterionic lipids, you form these very nice-looking sort of round structures which actually co-localize lipids and peptides. And these have all the sort of properties you might expect of condensates. You can dissolve them with hexane diol, you can do FRAP, etc. But if you wait for long enough, what you can see is you can see these types of filamentous structures growing out of these condensates. And it turns out that actually these are beta sheet rich, and these really have all the characteristics of the amyloid fibers that you would form from this peptide and that you would find in vivo in the case of people who suffer from diseases connected with aggregation of this peptide. So we then sort of asked, well, what is thermodynamically the relationship between these two phases, the liquid phase and the solid phase? Let's try and figure out the relative stabilities, and you can do this in a very sort of straightforward way. You just look at the critical concentration that you see in equilibrium with either the liquid phase or the solid phase. So this is what we get for the liquid phase under these kind of conditions. And if you do the same kind of experiment for the solid phase, you see that it's slightly more stable. So the concentration goes down a little bit, it's slightly more stable. There's not a massive thermodynamic driving force towards the solid, but it is more stable. And I should say this value here is, by the way, roughly consistent with the value that you get if you carry out this aggregation process without having a condensate there. So it looks like you end up in the same place regardless of whether you have a condensate. So the solid phase seems to be more stable for the system. How about the kinetics? So when I nucleate this solid phase, I obviously have two pathways. I can either go through from dilute solution or I can go through the condensate pathway. So which one is going to have the bigger flux? So the way to evaluate that, of course, is to compute the transition state energies. And so you can do that in a variety of different ways. But even based on dimensionality, it will be rates are always sort of this prefactor which contains some information about mass balance times this Boltzmann factor which contains information about the activation energy that you have to go over. And then here, obviously, this prefactor contains things like the concentration, which we can measure easily in the dense phase, the diffusion coefficient, which again we can measure from FRAP experiments and others. So we can actually compute all of these things to a reasonable level of accuracy. And if you do that for both of these pathways, this is sort of roughly what you find. You find that there's a significantly larger barrier. If you go straight from the monomer to the solid phase, there's a significantly larger barrier than either the barriers going from monomer to the condensate or from the condensate to the solid. So what that tells us is under many conditions, the nucleation, the pathway, the flux that leads from the monomeric peptide to the fibrillar state will be significantly enhanced if you can go through this type of condensate related pathway. So that's interesting. So if these states are metastable, if for many systems, if these liquid states are metastable relative to the solid, what that means is if you disturb the liquid state, it should relax into the solid state. So how can you perturb a single condensate? So we designed this type of microfluidic device where there's a very thin channel here, which allows us to change locally the solvent conditions, and then we can form individual condensates here. So the data here I'm showing is with a different protein. This will be with FUS. So let's see what happens if I perturb this condensate. So you can see initially it's clearly liquid, so you can see it flows, so this looks like a viscous liquid. If you let go of the pressure, though, something interesting happens, and let's play that again. So you can see here, initially clearly liquid, it looks like a viscous fluid, but when you let go of the pressure, then it bounces back. So that's clearly not something which you would expect liquids to do. So what is that? That's effectively elasticity, and in particular, it's a low-frequency shear modulus, which has appeared somehow in between the beginning and the end of the experiment, which of course is a characteristic of solids. So another characteristic is the fact that we have quenched internal dynamics, and you can see this very clearly in these FRAP experiments. If I take the system before shearing and I bleach in the middle, I have very rapid recovery. If I apply some shear, I have absolutely no recovery on the scale of the experiment. So it really looks like with this relatively minimal perturbation, over the course of a few seconds, these types of materials can go from a liquid to this type of gelled state. And it's actually interesting if you ask the question, how much shear do you need? It's really a very minimal amount of shear. And in fact, it's comparable to the level of shear that you might have in the intracellular space, especially if you have active transport processes. In some sense, it's almost slightly worrying. It looks like biology is using these condensates systems in cases where they're almost at the sort of verge of undergoing this type of irreversible transformation. It turns out, of course, if you leave these systems for long enough, they will undergo this type of transformation anyway on their own. A really simple way of measuring this is simply to take a system. This is continuing with FUS. So we take this system here, which sits somewhere here in this two-phase region here. And if I now go up, so I change the ionic strength in this case, and I cross the liquid-liquid binodal here, what happens is that obviously this condensate disappears. But if I now wait for some period of time, this is done at slightly elevated temperatures, so it's sort of accelerated aging. If I wait for 72 hours in this case, and I do the same experiment, I go from here to here, what happens is the condensate doesn't disappear. What does that mean? It means that it's no longer in the same phase. So if it was still in the original liquid phase, then obviously I cross the liquid-liquid phase boundary, it should dissolve. So it's in a different state. And so this is really, we're seeing this solid phase appearing here with a simple experiment. You can then go on and test for the mechanics, and you can actually show that this indeed has this low-frequency elastic modulus. But what's interesting is if you now do this experiment at intermediate aging times, you find something quite interesting. So, for example, after 24 hours, if I now try and cross the liquid-liquid binodal, what I see is that almost all of the condensate dissolves, but not completely. So I have sort of this rim of material here on the outside, which still remains here even after I cross this liquid-liquid binodal. So what is that? Well, this is this solid phase, which is starting to grow really on the outside of these condensates. And if you wait for longer, then it sort of starts to occupy a larger and larger space inside this condensate. And this is sort of an observation which I think many other groups now have also made for different systems. So this seems to be really a very sort of active part of the condensate, the interface between the dense and the dilute phase. That seems to be certainly for many of these systems where these nucleation processes take place. So I think when we look at these kind of sort of results, it sort of really becomes very clear that these macromolecular systems is really a fruitful way of thinking of their state by analogy to phase behavior of, say, small molecules. And of course, the way to understand phase behave, those kind of systems, is to generate phase diagrams and ask the question, where do I have a solid form, where do I have a liquid form, where do I have a gas form? But of course here for these systems, the sort of key parameters that we want to vary are sometimes temperature, typically not pressure, and more commonly just chemical composition. So what that means is in order to generate these types of phase diagrams, or the analogs of these phase diagrams, we have to generate lots and lots of copies of the system, but with slightly different composition. And so these are experiments which we initially started doing in microwell plates, and we made lots of different, pipetted lots of different combinations. None of the students liked doing those experiments because they said, I think quite rightly, they were boring experiments. So we spent a bit of time actually thinking about how we could do this in a higher throughput and slightly more automated way. And we'll be doing this using droplet microfluidics. So the setup looks like this. So these are devices which we make in our lab. The characteristic dimensions are sort of microscale. So this is roughly the dimension of a human hair. The video is slowed down roughly by a factor of 1,000. So this happens really very fast. You can make 1,000 microdroplets per second. And each one of these will have a slightly different composition. So I'm illustrating this with GTBP1 and RNA. And we can vary the flow rates. We keep the total flow rate constant, but we vary the relative flow rate. So each one of these droplets will have a slightly different composition. And then we can go in and look at these droplets using fluorescence microscopy, and you can see some of them, they will have condensates in them. So this droplet here is under conditions where the two-phase state is stable. This one here is under conditions where the single phase is, the homogeneous solution is stable. Then we have a neural network which looks at these and classifies them into two-phase or one-phase. Then I have one more problem left, is I have to, all these microdroplets, they all look exactly the same. So how do I know which one is which? How can I remember what was inside? And so what we do is we have a sort of trace amounts of inert fluorophores which encode the concentrations here, and then I measure that at the same time in 4 different colors, and then by doing that for each droplet I can figure out what its original composition was. And that's sort of what we're seeing here. This is roughly real time, so this really happens pretty fast at hundreds or thousands of drops per second. And these channels here I use to figure out the composition, and this one here I use to figure out the phase. And then what we can do is we can generate sort of data like this, where each one of these dots is an individual sort of measurement where we've mixed these two components in a given ratio. We color them depending on the phase they're in, and we place the points in X and Y based on the composition. And you can see here this very, very sharp phase boundary appearing as you'd expect for real thermodynamic phase transition. Based on what I just showed you, you might say, well, okay, this is clearly some type of dense phase, but how do I know that this is a condensate? Maybe it's solid. How can I tell whether this is a liquid or whether it's solid? And so what one can do is one can actually say, well, why don't we repeat the same experiment that I showed you in bulk for FUS? Why don't we just try and cross the liquid-liquid binodal inside each one of these drops and then see whether the condensates are still there? If they're still there, well, by definition, they're no longer in the same phase, otherwise they would have dissolved. So this is sort of the idea. So if I find some way of, for example, diluting each one of these microdroplets in order to cross the liquid-liquid boundary, the ones which disappear, well, they were still liquid, and if there's any ones which remain, those will be in a thermodynamically different state in the more stable solid phase. So how do I do that? Well, for the liquid-liquid boundary, I have my, we call this phase scan, so this approach I just showed you where we make lots and lots of different droplets, hundreds of thousands of different droplets, each one with a different composition. But now what I can do is I can take each one of these droplets and I can dilute. And so the way we do this is we inject into each one of these droplets on chip. You can see here the droplets arriving, and then here what we do is we inject some buffer into each one of these droplets. And again, this is slowed down roughly by a factor of 100 in this case. You can really do this at very high throughput, so you can easily dilute hundreds of thousands of droplets relatively easily on chip. And then what we do is then we ask the question, well, let's rerun them through this analysis system and see whether the condensates are still there or not. And you can see some of them seem to have disappeared. So here, this was in an area where there were condensates before, now there's none. These ones here still survive after dilution, so these ones are probably solid. So if I do this in a slightly more systematic way, I can change the time I give these condensates to age. So if I give them, this by the way is data shown for the protein tau. So on one of my first slides I showed you A beta, that's the protein that aggregates in Alzheimer's disease in the extracellular space. Tau is the protein that aggregates in Alzheimer's disease in the intracellular space. And there's actually increasing evidence that tau aggregates start life as liquid condensates. So if I give these tau condensates under an hour to age, the liquid-liquid boundary looks like that. Now I dilute all of the drops, and then I look at how many survive. None of them survive. So after 40 minutes, they're all still liquid. I dilute them, they're all gone. But if I wait for slightly longer, you can see some of these droplets, they're no longer dissolved, even if I try and dilute them. And the longer I wait, the larger the part of phase space is where these droplets no longer dissolve. So that's interesting. So by looking at this, now what I can do is I can, for each point in phase space, I can generate an aggregation sort of dynamics for each, in each part of phase space. I can see how long it takes to lose reversibility to go into the solid phase. So I get hundreds and hundreds of curves like this. And if I do a heat map, it looks something like this. So this is looking at the half time for this conversion. And it turns out that it depends where you are in the phase, in the two-phase region. So it turns out that there isn't one time scale for converting from a liquid to a solid. It really depends where you are. And here you can see it looks like the deeper you go into the two-phase region, the faster this liquid to solid conversion is. The other feature which is associated with these protein aggregates connected to disease is a structural transition. So they end up being beta sheet rich. So we thought it would be fun to try and see if we could understand whether these condensates, so we figured out a way to measure the liquid-liquid boundary, figured out a way to measure reversibility. We thought it would be fun to find a way to actually measure whether they contain beta sheet. So how do you do that? There's different ways of doing that. So we've adopted this kind of approach here, where there's dyes which intercalate into or attach to beta sheets and change the fluorescence emission spectrum. So this is doing this with a red emitting amitracker dye. And so then the idea is to do exactly the same experiment, except now I'm measuring the channel of that fluorescent dye, and then I can see whether that's beta sheets or not. I do the exact same experiment. I do my phase scanning experiment with this dye. And what I see here is I see something slightly different. So I see, as a function of time, this is the aging of the tau aggregates. I see a region here appearing pretty rapidly, which is beta sheet rich, and the longer I wait, the bigger the part of phase space is where I see beta sheet rich structures. And again, I can make some dynamics analysis for the rate at which this beta sheet appears in each one of these condensates. So, but now you could say, okay, well, we found a way to measure reversibility, we found a way to measure beta sheet. Are those correlated? Does that happen exactly at the same time? Is the only way you lose reversibility is by forming a beta sheet? Well, it turns out that it actually looks more interesting than that. So if I overlay both of these things, it looks like the structural transition is decoupled from the loss of reversibility. And in particular, if you look at the early times, everything is still reversible. So if I dilute, I recover the homogeneous solution, but I nevertheless have this area here, which is beta sheet rich. So this is a reversible beta sheet. So this is not yet sort of the fully formed amyloid structure. This is a reversible beta sheet. If I wait for slightly longer, I see this intersection here appearing of things which are irreversible and beta sheet rich. But this part here has lost its reversibility as well, but hasn't yet converted into a fully beta sheet rich structure. If I wait for long enough, then everything goes into sort of aggregated and beta sheet. So it shows that the sort of starting points, I think, are the ones that we in the field sort of thought. So we have an unstructured state, and it ends up being solid, irreversible, beta sheet rich. But actually, there's multiple pathways in which you can go through this phase space, and crucially, it depends where you are in the liquid-liquid sort of phase diagram, which one of these fluxes is most relevant. So that's interesting. If these solid aggregates start life as liquids, there might be a way to try and bias or at least prevent their formation. And this obviously is a big sort of area that many people have been very interested in from a therapeutic context, trying to find molecules which prevent aggregation, reverse aggregation. The only sort of successes which have been shown with reversing aggregation are effectively full-length antibodies for extracellular aggregates like A-beta. And the way that they reverse the aggregation is actually by recruiting active cellular clearance mechanisms. So the full-length antibodies, they still have the effector function. So it's really an active process. So it might be quite hard. So anybody who's tried to do this with small molecules has really struggled. So it might be quite hard to do reverse these aggregates once they're formed because of the very high level of sort of intermolecular interactions. But what we thought is clearly the liquid state, if that's a precursor, maybe by biasing the liquid-liquid phase boundary, maybe we might be able to sort of reduce the rate at which we end up forming the solid. So I'll show you how we've sort of gone about doing this for this protein here, FAST again. And so what we thought we would do is we would do sort of effectively a screen, but on the base of the phase behavior. So we're going to go through small molecule chemical matter, and for each molecule, we're going to measure a phase diagram. So what I'm showing you here is, in some sense, a repurposing screen. So we've gone through every one of the, I think there's about 3,000 or so molecules that have been approved by the FDA. So we took each one of them and we measured a phase diagram. And so then, again, the phase diagrams are measured in the same way as before. So you can see each one of these is an individual microdroplet with a particular composition, and you can see some of them have condensates, some of them don't have condensates. And then we look at this phase boundary. And so we went through the FDA library, and what we did is we found that most of the molecules don't do anything, so they sit here. But we found molecules which actually shifted the phase boundary both ways, which is sort of interesting. So these are small molecules, not an antibody, just a small molecule, and you can actually, in a relatively potent way, shift the phase boundary either way. You can make things where there's the two-phase part of the phase space is larger or smaller. And this is sort of what it looks like if you do a dose response. Obviously, these techniques are very flexible because you can sort of design what you want just by varying the pressures on the chip. You can design what you want in these phase diagrams. You can do a dose response. So this is sort of a DMSO control. It's flat, and this is, for example, what a hit would look like. You can see it gets rid, in a dose-dependent way, gets rid of the two-phase part of the parameter space. So that was in vitro system. Do any of these work in cells? So it turns out that of all of the FDA-approved drugs, we looked at each one of them in terms of the phase diagram. We found 17 which shifted the phase boundary in a significant way. It turned out that a good fraction of those, about 12, actually did the same thing in a cell model where we express FAST and we tag FAST. You can see here the condensates dissolved also in the context of a cell. You can go one level further. You can look at sort of simple animal models. So, for example, these are worm models here done together with Peter Hislop's lab. This particular model is expressing a form of FAST which is associated with one of the rare familial forms of ALS. And so what happens is these worms obviously accumulate aggregates. So if you look here at this worm, this wild type, this one here has a very significantly enhanced fraction of insoluble FAST. If we now add this molecule which shifts the liquid-liquid phase boundary, so this is a molecule which decreases the path of parameter space where I have the two-phase liquid, it pretty significantly reduces the overall aggregate burden in this worm. So it's sort of doing what we wanted it to do. So we're reducing the solid content by affecting the liquid-liquid phase boundary with a small molecule. And it turns out that these worms actually even exhibit sort of a motor phenotype. So because of the increased aggregation load, they're less able to wiggle around. So you can measure this, you can measure the thrashing rate, you can see this reduction here. And if you add the molecule, because of this reduction in the aggregate load, you actually even rescue a good fraction of this sort of motor phenotype. So we think this is sort of an interesting way of approaching these protein aggregation diseases, because it's been very, very hard to sort of go after the endpoint aggregate. So they're really very, very stable, and it's thermodynamically, if you sort of think about it in terms of the physical chemistry, it's probably going to be really hard to reverse the formation. Whereas we think that going slightly earlier, looking at these partly reversible states, it might be an easier sort of task to have that kind of effect. So obviously one of the sort of real questions I think that we and many people here have is why do some of these proteins seem to be very prone to form these phase separating compartments, and others seem to be less prone? What does it depend on? Does it depend on whether systems are homomolecular or not? So the way we sort of approach this is by trying to sort of look at this in a two-stage way. So we initially said, let's try and learn what are the physical features which give us separation, give us a sequence which is prone to phase separate when it's found on its own. And so you can do this in a variety of ways. There's many papers published on this. This was our sort of attempt from about 5 years ago. And so effectively what you can do is you can have specific physical features, the usual things like Shannon entropy, charge, blockiness, etc. And then you can have a language model where you pre-train on the human proteome, then you fine-tune on this specific task of trying to predict protein-protein aggregation, protein-protein interactions in condensates. And then it turns out that if you take what's quite nice, if you take a consensus model, it turns out the language models learn some features, the physical models learn some other features. You take a consensus model, you actually get pretty good performance in predicting whether proteins form condensates on their own in an in vitro system. So we then said, okay, well, let's now try and take a slightly more complicated system. Let's look at data sets where this process happens in a cellular context, and there's quite a few of these data sets around now, and just showing you one of the sort of early examples. It turns out that this model, which performs fantastically well in an in vitro setting, doesn't really work as soon as you try and look at things in a cellular context. So it has performance here, area under the ROC curve of 0.6 maybe, only slightly better than random. So that's interesting. So many of the proteins, a big fraction of the proteins which are found inside condensates, they're not there because of the intrinsic phase separation propensity. They're there for some other reason. So why are they there? So we thought, well, maybe it's because the proteins which are found inside condensates in a cellular context, maybe they're just promiscuous proteins. Maybe just like interacting with all other proteins and they just get sort of dragged together into this kind of cluster. So you can look at that. You can ask the question, if I split out proteins into two sets, the ones which are found in condensates, the ones which are not found in condensates, and then I ask the question, do they have more interactors in the full human proteome? You can look at StreamDB or some of these resources which summarize known protein-protein interactions, and it turns out that there's not really a big difference. Maybe with a bit of imagination, maybe they have slightly more interaction partners, but really not a big difference. So it's not that proteins which are found in condensates are just generally promiscuous and just interact with everything. However, if I now repeat the analysis and ask the question, the proteins which are found inside condensates, do they have more interactions with other proteins which are found inside condensates? There's a massive enrichment which is not there for proteins which are not found inside condensates. So it looks like there's really, and you can do a very similar thing with RNA. And if you do that, then now you have obviously much better ability to predict proteins inside condensates. This is what happens if you include just known protein-protein interactions on top of the physical chemistry. So this is simple phase behavior from the previous machine learning model. This is adding protein-protein interaction. You get even slightly better performance if you add protein-protein interactions and interactions with RNA. So this really tells us that the sort of physics of why proteins are found in condensates really separates into two very different origins. There's the intrinsic phase separation propensity, and you could call these proteins maybe, scaffold proteins. And then there's ones which are there, not because they're close to phase coexistence in any kind of statistical mechanics sense, but they rather just interact with proteins which are already found in this compartment. That's really what this seems to show. And if you now ask the question, well, what has this model learned? What are the sequence features which are enriched in these proteins? It turns out that you don't necessarily have the same things that you would expect for phase coexistence. So, for example, IDR fraction does come up, but it's not one of the sort of top features. Shannon entropy comes up slightly for non-RNA binding proteins. It's not really relevant for RNA binding proteins. And what you find is electrostatics comes up, in particular for RNA binding proteins, which obviously you might expect cation fraction to be relevant for condensates which contain large amounts of RNA. So it's really learning a completely different type of sort of sequence code when you ask the question, how do things get recruited into pre-existing condensates? If I now run this deep learning tool on the full human proteome, what I find is I don't find that everything is prone to form condensates. I find sort of really well-defined populations, but I do find sort of a tail at very high propensity to be found inside condensates. And you could ask the question, well, what exactly is in this tail? So what are these systems here which are in this tail? Is that real? And so what you can do is you can just in an unbiased way run this on the full human proteome, and you can say, well, let's have a look at the top four predictions. Let's see what they are. And then we looked in the Human Protein Atlas, which is really a fantastic resource for this kind of thing. Really contains so many different cell lines, so many different proteins stained with different antibodies. And what you find is you find all of these top four hits. So this is the protein stained in this magenta color here. And you can find for all of these four hits, indeed, if you look in the Human Protein Atlas, you can find that indeed they are present in the form of condensates. For the bottom four, none of them it shows any kind of condensation propensity. So we think that this is really now a tool which really summarizes both the intrinsic phase coexistence propensity, but also how interactions with other proteins and RNA can lead to proteins being recruited into these membraneless compartments. And these types of data really show that most of these systems are are really not homomolecular in a biological context. They really have multiple components. So the last thing I'll share with you is our attempts to try and analyze in vitro the strength of interactions, the composition of these condensates when they have more than one component. And so the approach that we've taken is to really try and think of whether we could measure tie lines, or at least approximations of tie lines. And so obviously the way this would work for a simple two-component system is I would prepare the system somewhere here. If I have two polymers, a blue one and a red one, which have an attractive interaction, I would prepare the system inside the two-phase region here, and it would spontaneously separate into a dense phase here. This is the condensate, and the dilute phase here, which lives here. And I connect these two, and this gives me a tie line. And of course, I could do this throughout this two-phase coexistence regime. And so obviously all of these lines by construction are ones which separate into so this line here, all of the points on this line separate to give me the same dilute phase by construction. And so why is that interesting? Well, it's obviously interesting because this contains information about the composition, but also the interactions, and you can see this in lots of different ways, including in this really simple sort of example where if I now switch the interactions from being attractive to being repulsive, I can also get phase separation, but now, of course, the nature of the dense phase is very different. So now this dense phase here is enriched in the red polymer and the blue polymer because I have an attractive interaction. This one here is depleted in the blue polymer but enriched in the red polymer. And now, of course, the tie lines change shape. So the slope of these tie lines also contains information about the collective interaction strength. So how can we measure this type of behavior at sort of reasonable throughput? Well, we go back to our droplet-based system, and what we do now is we pass each one of these droplets through a single molecule confocal here, and we measure the dilute phase concentration. And so this looks something like this. So here what I can do is I can really navigate around in phase space by just changing the pressures on the chip. So it's really a very easy way to move around in phase space. So if I sit somewhere here outside of the two-phase region, what happens is the data look like this. So each one of these is a microdroplet. You can see the dilute phase is the same everywhere. If I now go inside the two-phase region, the signal looks slightly different. So I can see these spikes here. So each one of those is an individual condensate. If I go further into the two-phase region, what I find is I find something interesting. I find the dilute phase went down. So I can get the dilute phase here, and I find more condensates. And so by doing these kind of measurements for each drop, I can now determine the composition, whether it has condensates, and what the dilute phase concentration is. And so then I can simply color. I can take a threshold, and I can just color drops with a given dilute phase concentration, and then obviously all of these droplets here by construction have separate into exactly the same dilute phase, and I can have a finer grid if I want. And so these are sort of really by construction droplets which all separate into the same dilute phase. So obviously in an n-component system, this is going to be an approximation to the tie line, and you can actually go through the statistical mechanics, and you can show, for example, that the sign of this slope is always the same as the projected n-dimensional tie line. You can give a bound on how accurate this is, etc. But for many sort of systems, this is really sort of a good proxy for the real projected n-dimensional tie line. And so now we can amuse ourselves by applying this for a number of different systems. I'll just show you one example, which is that of changing pH and asking the question, where do protons partition? Or do they partition, and which way do they partition? And so what we did is again, you can take a phase separating system. This actually turns out, this data here for an insulin, modified insulin peptide. We've done this for many different systems. This is about 75,000 droplets, where each one of the droplets has a slightly different concentration and a slightly different pH. And so then what we see is we see here a two-phase region here, and you can see the dilute phase there. It looks like the maximal extent of phase separation is roughly around the isoelectric point of the protein, so that makes sense. Proteins are minimally soluble around the isoelectric point, so we knew that. But what happens now if I look at these tie lines? If I ask the question, let's draw these lines with constant soluble phase concentration, what I can see is they're not flat. So you can see here, this has this kind of slope here. So what this means is that protons, there's a proton gradient. So the protons are not distributed uniformly, even though there's no membrane. It's water inside, water outside, but it's a gradient. And if I approach this two-phase region from the acidic side, the protons are actually depleted in the dense phase. So they're sort of, in some sense, ejected from the dense phase. If I do the same analysis on the basic side, I find the opposite slope. You can see here the opposite slope. So here the protons are sequestered into the dense phase. And you can actually make yourself an electrostatic argument for why this probably should be the case, because what happens is you minimize, these are very dense systems, you minimize electrostatic repulsion by being close to sort of dielectric point of these systems. And that's really enough to actually overcome the entropic penalty of having local proton gradients. And this can actually give a pretty significant effect. So this can be several pH units if you convert this into a pH gradient. So a very significant pH gradient, which is sort of interesting because in biology, if you want to have a pH gradient and you have membrane-bound compartments, you need to spend a lot of energy pumping all the protons out again, which leaked in. Whereas here with these membraneless compartments, it looks like you can generate pH gradients at equilibrium without having to spend energy pumping things backwards and forwards. And we think this may explain one of the reasons why, with all the mechanisms that biology has to compartmentalize things, why do you need yet another way of making compartments? But this might be one of the reasons because it allows you to create chemically distinct microenvironments, for example, even to the extent of having a different pH. And consistent with that idea, if you look at the isoelectric points of the subproteins associated with membraneless organelles, they have a very, very wide range of different isoelectric points, and therefore you'd expect them to have very different chemical microenvironments. All right, so those were some examples of sort of trying to look at this condensation phenomenon from a sort of physical chemistry point of view by generating sort of these large data sets. And our sort of view more generally is, I think when we go from, thinking about protein science, when we're going from individual molecules to molecular systems, I think this is really the length scale on which biological functionality emerges. So I think it's a really exciting opportunity to work at the systems level. Obviously, at this level, the systems will be more complex, and we think the more complex the systems are, the bigger the advantage is if we can generate more data. So we're very excited about these next generation experimental approaches where we can generate larger data sets. This was all work done by my fantastic group of PhD students and postdocs in Cambridge. Lots of it was done in collaboration with people either locally or worldwide, and thanks to the people to fund our work, and thanks for having me here.
That was very inspiring. I have a comment and a question. In 2021, we wrote a paper about homotypic, heterotypic phase separation and the generic features of the tie lines. And we showed that depending on how the noise in the protein concentration is aligned, there's some distribution of concentration, how that noise is aligned with the tie lines tells you how effective buffering of the noise is, and I can send that to you. So that might be something very interesting to look at in your many, many drop experiments, how noise and the tie lines are related. The question that I have is, at the beginning you talked about a lipid that you put stable. Is that true in all the droplet stuff that you showed us, or only at the very beginning? And does the lipid affect somehow what happens if you play with let's say just the lipid concentration at the surface? Does it affect fibril formation, etc.?
Yeah, so that's a great question. So the data I showed, it's only the A beta example which contained lipids, but inspired by sort of that finding, we're now increasingly looking at the role of lipids in condensate formation more generally. And it's actually interesting, we're finding many systems, I don't have the data here, but we're finding many systems where actually you do form these mixed sort of systems which contain both protein. Obviously, we knew protein and RNA co-localizing condensates. We increasingly think that there can be many other components as well, including lipids. The interesting thing with A beta is it doesn't really form these kind of structures in the absence of lipids. And so that's why those data were shown in the presence of lipids. It's sort of really interesting because you can see this in very simple experiments. If you just take it forms on many different membrane compositions. It even forms on cellular membranes. And these are, I think, experiments that many labs have done. If you just take soluble A beta and you put it on top of many different cells and you look at the cells, you do see sort of round-looking clumps on the outside of the cell membranes. And I think, this is something which nobody's really reported or looked into. But obviously, if you then dig a little bit deeper, what you find is these are indeed mixed structures, and they have sort of very much the features that you would expect from complex quasivets or condensates.
Nice talk, thanks. So in the multi-component tie lines, they were less sharp than the other systems. Is that due to uncertainty in determining the concentration, or is there like an inherent metastability that is sensitive to other things, or?
So I think there's lots of really interesting points in what you're saying. So I guess there's sort of a question of how sharp is the phase boundary, and then how sharp are the other tie lines. And at some level, I think a lot of the sort of fuzziness of the phase boundaries is actually experimental error, and it's actually it comes from two sources. It's first of all misclassification of the phase, and this is something it's sort of fascinating. We sort of switched to doing this with deep learning rather than sort of conventional computer vision techniques probably 4 years ago. And interesting, the performance went through the roof, so that's sort of really when it became clear that was the way to do this kind of analysis. So that error is now reduced, but we still have some error on actually positioning the points, and that gives a bit of fuzziness. I think most of the stuff that you're seeing here in terms of the error on the phase boundary is actually experimental error. So as we go along, more statistics, we get better and better at reducing that error. But that's sort of interesting on its own, because you might have expected there to be some real statistical mechanics. So, for example, you might have expected there to be rare nucleation events, etc. We see this when we look at the solution to solid transition. We see very significant sort of statistical mechanical number fluctuations and really intrinsic stochasticity. But we don't really see that when we look at the solution to liquid transition, which is interesting.
Yeah, cool. Thanks.
Hey, the microfluidics experiments are beautiful, really beautiful.
Thank you.
I had a question in something that you've been showing in the very beginning, the delta G plot as a function of the species that you get. How are they being estimated? Like the delta G forming a condensate or forming the solid?
I see. So this is, for example, when I showed the data for the A-beta peptide. Yeah. So I mean, effectively, what we do is we measure the concentration of the dilute phase in equilibrium with each one of the phases, and effectively, if the solid or the liquid phase has a large sort of volume, large number of molecules, then to a good approximation, the free energy difference will be related to the log of the concentration of the soluble phase.
And I guess when discussing the stability of the solid phase or the condensate phase, how accurate is this delta G prescription? For instance, you're saying that this formation of the solidified aggregate is irreversible in the sense that when you add salt, it doesn't dissolve, but the condensate does dissolve. Like, is that just raising the delta G of forming a biocondensate, like maybe lowering the monomer, but leaving unaffected the aggregate, or?
Yeah,
is that
that's a fantastic point. So in some sense, there's almost sort of two different questions. So the first one is how accurate can you determine using this method the delta G, and how is the delta G related to reversibility? So for the measurement, it turns out that even though it's in principle a trivial measurement, what we've learned over the years, we've been doing these measurements for many years now, largely for the solid phase and more recently for the condensate phase. It's actually not so easy to do because you have to be sure that you get the equilibrium concentration. And so the easiest way to show that is if you approach equilibrium from both sides. So if you dilute and you see if you establish the equilibrium concentration, you add monomer and you see if you get the same concentration, then you have some confidence that you're at equilibrium. So sometimes the concentration measurement itself is easy, although sometimes the concentrations can be low if the stability is high, but that's in principle easy. The hard part is to make sure you're really measuring the thermodynamic equilibrium concentration. But that's something that can be done. How is that related to stability? I mean, I guess the reversibility, so how is stability related to reversibility? I mean, the reversibility, I guess, is related to the height of the barrier going the other way. And so I think it's partly the increase in stability, but I think it's also the fact that the nature of the interactions is different. So the rate at which you can dismantle, for example, so many of these end up as being these dense beta sheets. And so that's something which is kinetically really hard to dismantle. So I think that's largely where the reversibility comes from.
Thank you.
Well, the expert setup, Bill was...
amazing talk as always. I'm a little confused on the same plot about how to interpret the barriers that you presented in the context of the kinetics that you showed later. Specifically, it appeared that the barrier from the dilute solution to the condensate was higher than from the condensate to the solid phase as you drew it. And that would suggest to me that the rate limiting step would actually be the first thing, and so then I wouldn't see a metastable condensate on long times. So I guess two questions. Should I be thinking about the prefactors being tremendously different if I take that plot literally? And secondly, is the second transition to the solid phase really a thermodynamic phase transition in your view, or something more continuous And so maybe this sort of barrier picture isn't really the right way of thinking about it.
Yeah. So when I start with the latter, so it's a great point. So when we're showing these, when we show the liquid-liquid phase boundaries, those are really thermodynamic phase boundaries. Obviously, there's experimental error, etc. But in terms of the physics, that's really the thermodynamic liquid-liquid phase boundary. When we're showing these liquid-solid phase boundaries, they're clearly not thermodynamic phase boundaries in any conventional sense. Why? Because they change as a function of time. So effectively what we're seeing there is really this kind of dynamic, I think it's more similar to glass formations. Effectively what we're seeing is sort of this kind of dynamic arrest, which seems to take place at different rates in different parts of the phase diagram. So it's not a thermodynamic phase boundary. At the end points, where if you wait for long enough, then the boundary you get for the dilute solid, that's probably a good approximation of the thermodynamic dilute solid phase boundary.
Okay, we are running out of time. Maybe we can talk with the speaker later. So.